Many research projects fail, not because the topic is bad, but because the design does not match the question. Research projects often fail before the first data point is collected, even when the underlying clinical topic is brilliant. A weak design acts as a hard bottleneck. It takes an urgent clinical hypothesis and renders it impossible to answer using the study's parameters. Conversely, a rigorous design protects the researcher. It shields against systemic confusion, isolates flawed data collection, prevents incorrect statistical analysis, and ensures final conclusions are valid. A study design functions strictly as an architectural blueprint. The specific demands of the research question dictate its structure, regardless of perceived prestige. A study design is the explicit plan for how a researcher will answer the what of their research question. This blueprint defines specific variables: the study population, the metrics used, the timing of measurements, and the presence or absence of an intervention. This diagram illustrates the core pathway. Moves sequentially: research question, purpose, time direction, exposure or intervention, comparator, and outcome. These steps automatically narrow the available options. The logic of the question forces the researcher toward the correct methodology. The final honest interpretation a researcher can make is bound by the pathway established at the very beginning. The blueprint determines the limits of the conclusion. To navigate these options, we use a design decision map. Choosing a design requires an explicit statement of purpose. When the goal is to describe a population without comparing groups, you conduct a descriptive study. This involves documenting traits in their natural state. For example, mapping the clinical characteristics of patients with diabetic ketoacidosis. They are simply cataloging present traits. Descriptive studies are highly feasible for beginners, serving as an engine for generating hypotheses. But this design has strict limitations. Without a comparison group, they cannot test causality and remain vulnerable to selection bias. Descriptive studies map exactly what is present in a population, though they cannot explain why those conditions occurred. If the research objective shifts to measuring how common a condition is at a specific moment, the purpose is prevalence. This leads to the cross-sectional study. Think of this as a simultaneous snapshot of both exposure and outcome at one point in time. Measuring resident burnout through a survey provides a clear view of what is happening now. However, notice how the exposure and outcome icons swap places. This is the temporality problem. Measuring everything at once means we cannot prove which variable came first. Cross-sectional studies map present associations effectively, yet they stay silent on cause and effect relationships. When an outcome is rare, capturing it in a snapshot is difficult. The researcher must investigate past exposures that differ between groups. This is a case control study. The structure identifies people with the outcome, the cases, and compares them to healthy controls by looking backward through their history. For example, researchers identify patients with inpatient falls and look back through records for previous hypoglycemia. While efficient for rare outcomes, looking backward introduces vulnerabilities. Self-reported data is subject to recall bias, and hospital charts to documentation bias. This design trades the ability to calculate true incidence for high efficiency when investigating rare clinical outcomes. To determine if a specific exposure leads to a future outcome, the researcher must track participants forward through time. In a cohort study, the population is grouped by exposure first, then followed forward to see who develops the outcome. This can be done prospectively or shifted into historical records for a retrospective cohort. Regardless, analysis always flows forward from exposure to outcome. Cohort studies offer the massive advantage of clear temporality. The main risks involve losing participants over time and the influence of unmeasured confounding variables. The cohort design remains the most authoritative observational method for proving exposure-outcome associations and calculating real-world clinical risk. To move from passive observation to proving a treatment works, the researcher must take active control of the exposure. In a randomized controlled trial, the researcher assigns the exposure through random assignment. This process ensures that both known and hidden confounding variables are distributed evenly across the groups. This allows testing if an education program reduces diabetic readmissions compared to standard care. Randomization provides the strongest support for causal inference, but has constraints. These trials are expensive, time-consuming, and face ethical barriers. Randomized trials provide the highest level of evidence for causality, though many clinical questions are not legally or practically suited for them. Many essential research questions exist outside the framework of tracking exposures over time. To test a new tool, we use the PIRD framework: population, index test, reference standard, and diagnosis. This is a diagnostic accuracy study. Evaluating D-dimer for pulmonary embolism requires comparing it to a CT scan. The accuracy of the study depends entirely on the quality of that reference standard. Qualitative studies shift the focus from numbers to human experience. These methods explore the why and how behind clinical behaviors. Qualitative research maps complex barriers and perspectives, prioritizing depth and context over statistical prevalence. Finally, systematic reviews and meta-analyses pool data from multiple existing studies to find an overall conclusion. A systematic review functions as a structured research study of existing literature. It provides immense value for decision-making, though it remains limited by the quality of the studies it includes, the rule of garbage in, garbage out. These specialized designs address critical clinical questions that raw numerical tracking cannot solve. Structural errors made during the planning phase can doom a study before the data even arrives. Common mistakes include selecting a design name before defining the question, mislabeling historical cohorts as cross-sectional studies, and claiming causality from observational surveys. True mastery of study design involves ensuring the study's purpose aligns with the mathematical limits of the data. Modern research planning now integrates artificial intelligence as a high-speed logic assistant. This technology accelerates the mapping process while leaving the final architectural decisions to the researcher. AI can be categorized by function: generative tools for comparing designs, literature tools for finding past methodologies, and source-grounded tools for querying specific guidelines. A researcher might prompt the AI to act as a mentor and compare the feasibility of different designs for their specific question. The most effective strategy is to force the AI to list limitations, bias risks, and data requirements for each option, rather than asking for a single answer. Safety red lines are essential. AI must not be allowed to invent citations, determine study feasibility without data, or justify causality from weak designs. AI can map the landscape of possibilities, but the final blueprint must be justified by human statistical judgment and ethical standards. A study design is only as strong as its alignment with the research question and the flow of time. To succeed, a researcher must match their purpose to the design, verify the time direction, map all potential biases, and ensure the final conclusion matches the authority of the blueprint. The most effective study design prioritizes methodological honesty and data alignment over sheer complexity. Full lectures, audio resources, and supplementary materials are available from the Bayyinah Health Research Advisory at the link on your screen.